welcome all the delegates and thank Dr. Heba Siddiqui, moderator, and Magnus Group organizing team for a brilliant platform today at Gynecology and Obstetrics Women's Health Scientific Sessions, Gynecology 2025, Orlando, Florida, United States of America, prestigious scientific sessions meeting. I'm Dr. Saumya Pandey, PhD Life Science, recently worked as head clinical research at leading fertility hospital, Indira-IVF, Udaipur, Lucknow, India. And the title for my talk is Impact of Matrix Metalloproteinases, MMP2 2735C2T, and Tissue Inhibitor of Metalloproteinase, TIMP2 2418G2C, gene polymorphisms with human papillomavirus-mediated cervical cancer, emerging trends in gynecologic oncology. Let's go ahead with the talk. May I request the technical support team for a smooth, seamless slide transition? Next slide, please. Thank you. Gynecology, women's health, snapshot. Human papillomavirus is the major etiological agent of cervical cancer, leading cause of morbidity and mortality in women worldwide. Screening strategies for reducing the burden of HPV-mediated carcinogenesis are emerging as effective means for cervical cancer control and prevention in developing countries. And I have highlighted this in my impactful publications as lead author in Gynecologic Oncology, International Journal for Gynecological Cancer, Molecular Biology Reports, and Asia Pacific Journal for Cancer Prevention, along with Journal of Community Health with cervical cancer control and prevention, cost-effective strategies as being adopted globally to reduce the disproportionate share of HPV morbidity and mortality in cervical cancer in the Gardasil vaccination era. Next slide, please. The anatomical location of cervix is well known, with ureters, uterus, the ovary, bladder, cervix, and urethra with human papillomavirus, primary HPV 16/18 as the major etiological factors leading to cervical carcinogenesis in women of varying genetic landscapes. Next slide. Host factors, including variations in low penetrance genes, may explain some of the inter-individual differences in cervical cancer susceptibility in women worldwide. And there comes the importance, the emerging significance, the innovative role of single nucleotide polymorphisms as the most common form of genetic variants in human genome, some of which have the potential functional influence on susceptibility to human diseases, including cancer, with emphasis on HPV-mediated cervical cancer and targeted manipulation of host genetic factors, the immunoinflammatory carcinogenesis mechanisms, including the complex interplay in the repertoire of toll-like receptors, matrix metalloproteinases, tissue inhibitor of matrix metalloproteinases, the cyclooxygenase 2, as well as other host genetic factors, and the therapeutic targeting of metabolic biochemical signaling cascades may lead to the eventual design and development of predictive and prognostic biomarkers for cost-effective management of cancer in women worldwide. Next slide, please. Immunobiology of toll-like receptors, the emerging trends. There has been a lot of emphasis on the therapeutic targeting of toll-like receptors, a repertoire, a family of evolutionary conserved pathogen recognition receptors as the pivotal primary sensors of invading pathogens that identify the molecular motifs of infectious agents, pathogen-associated molecular patterns, and elicit an effective defensive response against them. There are 10 TLRs known in humans, deriving their name from Drosophila toll proteins, and the complexity leading to the activation of immunoinflammatory cytokines, chemokines, NF-kappa B with the TRIF-mediated pathway and the MyD88-mediated pathway. So the targeted manipulation of toll-like receptors may lead to the eventual design of predictive and prognostic markers in HPV-mediated cervical carcinogenesis in the Gardasil vaccination era. And this is one of my first publications, Saumya Pandey from Biomedical Sciences, Creighton University School of Medicine, Omaha, Nebraska, United States of America, published on August 6, 2006 in prestigious Immunology and Cell Biology. So a lot of collaborative, globally enriching research has been in the last few years, or I should emphasize in the last 2 decades, with emphasis on the enigmatic array of toll-like receptors in conjunction with apoptosis, autophagy cell death-mediated pathway, and the Matrix Metalloproteinase and the tissue inhibitor of Matrix Metalloproteinase, TIMP2-mediated pathway, to dissect the complexities, the intricacies involved in host genetic factors and the causal factors leading to HPV-mediated cervical cancers in women of varying population pools, of varying lifestyles and genetic landscapes. And this could also be the next step for global collaboration in oncofertility, gynecologic oncology, fertility, preservation-mediated women's health research studies for the eventual design of precision oncology-based medicine with emphasis on overall evidence-based, patient-friendly women healthcare globally. Next slide, please. The TLR signaling pathway, as I just discussed, a complex repertoire of TLRs, TLR 1, 2, 3, 4, 5, 6, 7, 8, and 9, 10, with the majority of TLRs activating pro-inflammatory cytokines and chemokines via the MyD88 pathway with intermediary kinases and leading to NF-kappa B upregulation, and the TRIF-mediated pathway with TLR3 and TLR4 working via the TRIF, leading to caspase recruitment, which has significance in apoptotic cell death, which has emphasis in carcinogenesis, and leading to the activation of type 1 interferons. So a lot of complex crosstalk in the TLR-mediated pathway in conjunction with various biochemical, metabolic, cell signaling pathways to dissect the intricacies, the underlying pathogenesis of HPV-mediated cervical cancer is being studied globally on a cost-effective, patient-friendly healthcare, women's healthcare management platform. Next slide, please. The human papillomavirus structure is indeed complex, and the regulation of virus gene expression and virus replication with p53 telomerases, retinoblastoma, disruption of cell growth cycle, the L1 capsid proteins, the L2, the E1s, E2, E3, and the DNA replication, frequently disrupted site in case of integration into the host genome, gene expression, upregulation and downregulation of immunoinflammatory genes, assembly and release of inflammatory genes with the nucleocytoplasmic shuttling, transcriptional activation, and the membrane signaling proteins coming into the play with few epigenetic modifications, and the causal genetic association needs to be studied. And this has been reviewed by me, Saumya Pandey, in a prestigious journal, Frontiers. Next slide, please. Schematic overview of risk factors in cervical cancer, HPV infection, clearance of HPV, the SIN1 primary nuclear abnormalities, including tetraploidism, regression, development of aneuploid cell clones, progression to the advanced SIN2 and SIN3, and cofactors could viral integration, smoking, hormones, immune status, other infectious agents, secondary genetic changes, other environmental factors lead to invasive cancer. And there could be a growth advantage of a cell clone with invasive properties. So a lot of immunoinflammatory crosstalk with the initiation of HPV infection and the recurrence of HPV and the clearance of HPV leading to the viral chronicity, the viral factors getting overexpressed, leading to early and advanced cervical carcinogenesis in women of childbearing age and of varying genetic landscapes that could even impair the fertility potential of the women of childbearing age. So this is a public health challenge, and we need to join hands today at the prestigious Gynecology Women's Health, Orlando, Florida, United States of America, meeting with all the delegates, all the participants, to team up for a global collaboration to address the emerging issues that lead to the distress, sometimes even financial hardships of women of varying genetic landscapes, of varying lifestyles, with all the malignancies. So we should join hands to design cost-effective, patient-friendly, evidence-based management strategies for reducing the burden of the ever-increasing complexities of gynecological malignancies, even HPV-mediated cervical cancer in women of varying genetic landscapes that could even impair the fertility potential. So a lot of thrust on oncofertility and evidence-based women's healthcare in the coming years. Next slide, please. Autophagy in cervical cancer pathophysiology has also been studied with emphasis on cellular stress with Beclin-1 and the ATG25712 and the LC3-1 and 2, which are the microtubule-associated light chain protein 3 proteins that lead to immunoinflammatory crosstalk mechanisms leading to aberrant physiological milieu, hypoxia, inflammatory cell infiltration, leading to HPV-mediated cervical carcinoma. Next slide, please. Tumor cells produce enzymes that destroy the matrix barriers surrounding the tumor, permitting invasion into surrounding connective tissues, entry and exit from blood vessels, and metastasis to distant organs. These enzymes could degrade the extracellular matrix ECM that have been viewed as essential for tumor progression, even HPV-mediated cervical carcinogenesis. And this is where the role of Matrix Metalloproteinases comes into play that degrade virtually all ECM components, and therefore classically MMPs were recognized as being produced and secreted by tumorous cells, degrading basement membrane and extracellular matrix components, thereby facilitating tumor cell invasion and subsequent metastasis, leading to aberrant cell proliferation in the hypoxia, inflammatory, tumorigenic cell physiological milieu, leading to carcinogenesis. Next slide, please. HPV-mediated cervical carcinogenesis reviewed by me, Saumya Pandey, in Frontiers, that there is a lot of thrust on protein-based and peptide-based vaccines, primarily Gardasil and Cervarix, the nucleic acid vaccines, whole cell-based vaccines, and live vector vaccines. So, apart from the causal genetic association with predictive and prognostic biomarker development and therapeutic targeting of biochemical cell signaling pathways, primarily TLRs, MMP2, TIMP2, and autophagy machinery, we need to even lay emphasis on the efficacy of cost-effective vaccination for mass screening of cervical cancer in susceptible women of varying genetic landscapes, of varying cohorts, of varying population pools, to reduce the disproportionate share of morbidity and mortality associated with HPV-mediated cervical cancer. Next slide, please. Objectives of my study were: inflammation is a hallmark of HPV-mediated cervical cancer, Matrix Metalloproteinase, MMP2, and Tissue Inhibitor of Metalloproteinase, TIMP2, are emerging as pivotal players in inflammation and carcinogenesis. My innovative public health-oriented study aimed to evaluate the role of MMP2 735C2T, reference sequence 2285053, and TIMP2 418G2C, reference sequence 8179090, gene polymorphisms in HPV-mediated cervical cancer susceptibility in North Indian women. Next slide, please. Methodology: it was a hospital-based case control causal genetic epidemiology study in Lucknow, Uttar Pradesh, India, with a power analysis sample size calculated by Quanta software with input of variables with significance level alpha less than 0.05, model of inheritance log additive, allele frequency lowest in controls, and genetic effect for odds ratio more than 1.65, yielding considerable high, more than 80% statistical power, with a study sample size of 400 study subjects, 200 histopathologically confirmed cervical cancer cases, and 200 healthy controls with age-matched and similar ethnicity North Indian. Genomic DNA extraction from peripheral blood samples collected from study subjects was carried out using the salting out method by Miller et al., and MMP2 and TIMP2 genotyping was performed using polymerase chain reaction-based restriction fragment length polymorphisms. The chi-square goodness of fit test was used for any deviation from Hardy-Weinberg equilibrium. In controls, chi-square analysis was used to determine the differences in genotype and allele frequency with binary logistic regression application for estimating age-adjusted odds ratio with Bonferroni's correction for multiple comparisons in few subgroups, where I did the subgroup stratification with lower sample study cohorts. Next slide, please. The case control study: 400 study subjects, North Indian ethnicity, cervical cancer cases 200, controls 200, blood sample collection, DNA extraction by salting out method, well demonstrated by Miller et al., PCR RFLP, genotyping, and data analysis using SPSS software. Next slide, please. The inclusion criteria for cervical cancer cases and controls: histopathologically confirmed FIGO stages 1, 2, 3, and 4, de novo cases without undergoing radiochemotherapy, North Indian ethnicity, pre-operative, and not on any radiation or chemotherapy regimens. For controls, the inclusion criteria was well-defined: age-matched, healthy, cancer, HIV-free females, randomly selected from general population, same ethnicity as the cervical cancer cases, no family history of malignancy, allergy, hypertension, diabetes, or cardiovascular diseases. Next slide, please. Exclusion criteria was well defined again for cervical cancer cases as well as controls in a statistically powered, more than 80% statistical power study, sample size of 400, as I had discussed, calculated via the Quanta software. Exclusion criteria: post-operative, presence of other gynecological malignancies, presence of hereditary genetic disorders, ethnicity other than North Indian, and presence of chronic clinical problem and or disease, presence of hereditary genetic disorders, ethnicity other than North Indian were the exclusion criteria for controls. Next slide, please. Causal genetic association of MMP2 TIMP2 polymorphisms with HPV-mediated cervical cancer in Asian Indian women of North Indian ethnicity. So I addressed the risk of developing cervical cancer in North Indian women, the clinical stages of cervical cancer, FIGO stages 1, 2 early versus FIGO stages 3 and 4 advanced. Modulation of cervical cancer due to environmental carcinogen tobacco usage, either in tobacco smoking form, in the active form of tobacco as cigarette smoke, or passive form with the partners of cervical cancer women, and the passive form of tobacco along with the chewing. So modulation of cervical cancer due to the usage of tobacco should also be kept in mind while emphasizing the causal genetic association for MMP2 TIMP2 polymorphisms with HPV-mediated cervical carcinogenesis in Asian Indian women of North Indian ethnicity, as I explored in my innovative public health-oriented causal genetic epidemiological study. And we can expand using multicentric, multi-cohort population pools of varying genetic ethnicities and lifestyles by replication of these findings in a multicentric, large population pool of women of British, American, Australian, African, and other regions with a mixed cohort of women for eventual design of patient-friendly, evidence-based, precision oncology-based, cost-effective, predictive, and prognostic biomarker development for HPV-mediated cervical carcinogenesis in women worldwide. Next slide, please. Results of my pilot innovative study demonstrated no significant association between MMP2 and TIMP2 gene polymorphisms and risk of developing cervical cancer in the study population. Nonetheless, interestingly, stratified analysis using the subgroup case-only study approach revealed no causal effect of MMP2 TIMP2 polymorphisms on early figure 1 and 2 versus advanced stages figure 3 and 4 of HPV-mediated cervical cancer. Therefore, MMP2 and TIMP2 polymorphisms did not modulate the risk in cervical cancer patients who smoked tobacco cigarettes in my innovative study. So there could be the role of environmental risk factors, host genetic factors, lifestyle factors that led to low significance, and we need to study a multicentric, multi-cohort population pool-based, a very large sample size study to eventually design biomarkers for MMP2 TIMP2 in conjunction with the enigmatic array of toll-like receptor gene polymorphisms for eradicating the disproportionate share of morbidity and mortality associated with cervical cancer. And interestingly, I have few impactful findings with toll-like receptor gene polymorphisms with emphasis on TLR2, 3, 4, and 9 in cervical carcinogenesis, leading to more promising results even with the modulation of tobacco-mediated cervical cancer. So lots of crosstalk in toll-like receptors, autophagy, apoptosis, MMP2 TIMP2 with evidence-based causal genetic association studies needs to be studied with a multicentric, a very large cohort sample size for the eventual design of patient-friendly, cost-effective HPV management protocols for eradicating the increasing risk, the increasing burden, the increasing distress, even financial distress associated with cervical cancer in women seeking clinical care and expert medical care with restricted financial resources. And there could be even the role of biobanking with fertility preservation in HPV-mediated cervical cancer women so that fewer younger women susceptible to cervical cancer in the early figure 1 and 2 stages or the advanced figure 3 and 4 stages may go for fertility preservation with biobanking for fulfilling the cherished dream of having a child, a fulfilling family, despite seeking treatment for HPV-mediated cervical cancer. So lots of emphasis even on oncofertility, public health, women's research. Next slide, please. The demography and clinical characteristics of cervical cancer patients as per classification of FIGO. The mean age was 48.27 plus minus 10.04 in cervical cancer patients 200 and 46.95 plus minus 9.81 standard deviation in controls. Stage 1, 21 patients, 2, 71, 3, 104, and 4 patients. Tobacco users, 55, 27.5%. Non-users of tobacco, 145, 72.5. So relatively low number of tobacco users in the present study. And in controls, tobacco usage was not taken because of the ascertainment bias. Next slide, please. Figo stages of cervical cancer, the characteristics of cervical cancer patients with the stratification of stages 1, 2, 3, and 4 in 200 histopathologically confirmed cases, and the tobacco usage in cervical cancer cases with stage 1, 10.5%, stage 2, 35.5%, stage 3 cases were a total of 104 out of 200, 57%, and a very lower number of advanced malignant stage 4 with 2%. Tobacco usage, 145 out of a total of 200, 72.5% tobacco non-users, and 27.5% were tobacco users. Next slide, please. Clinical stages of cervical cancer, stages 1, 2, 3, and 4 were diagnosed in 21, 10.5%, 71, 35.5%, 104, 52%, and 4, 2% patients respectively, while the early stages were clubbed stages 1 and 2 FIGO, and the advanced FIGO stages 3 and 4, with stage 1 and 2, total 92 cases, 46%, and advanced stages, clubbing stages 3 and 4, FIGO stages 3 and 4, 54%. That was a total of 108 cases of HPV-mediated cervical cancer in my study with 400 study subjects. Next slide, please. MMP2, also known as 72 kilodalton gelatinase or type 4 collagenase, is a member of the MMP family that primarily hydrolyzes gelatin and type 4 collagen, the major structural component of basement membrane, which also has an activity against a spectrum of other proteins, such as the growth factor binding proteins and growth factor receptors. Furthermore, the production of this proteinase in tumors is not only by cancerous cells, but also by normal stromal cells. And further, the activity of MMP2 amongst other MMPs is also regulated by endogenous factors, including a family of antiproteinases known as Tissue Inhibitors of Metalloproteinase, TIMPs. And of the 4 members of the TIMP2 family, TIMP2 is particularly interesting because of its dual functions in terms of enigmatically regulating MMP2 activity and its paradoxical effects on certain cancers. So a single nucleotide polymorphisms -418 G to C has also been identified in the promoter of the TIMP2 gene, which can be therapeutically biochemically targeted for causal, genetic, host genetic, epidemiological association studies for the eventual design of evidence-based, cost-effective, predictive, and prognostic biomarkers in risk stratification of HPV in users of tobacco and non-users of tobacco with HPV high risk and HPV advanced stages FIGO 3 and 4 of cervical cancer. Next slide, please. Although the functional significance of this germline polymorphisms is currently unknown, downregulation of transcriptional activity due to the variant has been suggested because of the G to C substitution, which is located within the consensus sequence of the SP1 binding site in the promoter region of TIMP2. And it is therefore reasonable to postulate that this polymorphism may downregulate TIMP2 expression and consequently cause an imbalance between the activities of TIMP2 MMP2, which is believed to have a significant impact on cancer development and progression. Next slide, please. Genotype and allele frequency distribution of MMP2 and TIMP2 polymorphisms in cervical cancer cases and controls, with a total of 400 cases and 400 controls, was promising, with the MMP2 RS2285053 CC genotype in 81.5% cases versus 86.5% controls, CT 17%, 12.5%, TT MMP2 genotype 1.5% and 1%. CT and TT genotype clubbing was 18.5% versus 13.5%, and the C versus T allele 90 and 10 and 92.8 and 7.2, with the p-values being promising 0.172, 0.659, 0.156, and 0.157, with the age-adjusted odds ratio and the confidence interval showing promising, innovative, public health-oriented studies with TIMP2 GG polymorphism genotype 150 cases out of 200, 165 in controls, 49 for GC, 35 for GC controls, CC genotype of TIMP2, just one, which is 0.05, and nil, none in the controls. GC and CC clubbed together, the genotype frequency being 50, 25%, 35, 17.5%, with 0.087 as the p-value, and the GNC allelic frequencies 87.3 versus 91.3 in cases and controls, while 12.7 and 8.7 in cases and controls for the C allelic frequency. So a lot of complexities associated with the genetic polymorphisms of MMP2 and TIMP2 in studying the causal association of HPV-mediated cervical cancer. Next slide, please. Association of MMP2 and TIMP2 gene polymorphisms with early figure 1 and 2 versus advanced 3 and 4 stages of cervical cancer was the CCMMP2 genotype 78.3%, while in advanced stages relatively higher 84.2%, CT genotype frequency 21.7% in early stages 1 and 2, while 13.0% in advanced stages with p-value 0.132, TT nil in early stages, and 2.8% in advanced stages. And total number of cervical cancer cases of early stages figure 1 and 2 were 92, as I had discussed earlier in the demography and clinical subgroup stratification, and advanced stages 3 and 4, 108, with the odds ratio and confidence interval level appearing promising, with the TIMP2 RS817990 GG genotypic frequency in the early stages 1 and 2 being 77.2%, while in advanced stages 73.1%. So almost similar, 21.7% GC and 26.9% GC for the advanced stages. So a slight tilt with the skewing of the GC TIMP2 frequency towards the higher side in the advanced club stages, while CC nil in the advanced stages. Association of MMP2 and TIMP2 gene polymorphisms with environmental carcinogen tobacco usage among cervical cancer patients was promising, with total tobacco users 55 and total non-users among the cervical cancer 200 cases 145, with the non-users CCMMP2 80%, 85.5% in the users, 18.6% for CT, and 12.7% for CT in the users. Amongst the non-users, the TT-MMP2 genotypic frequency was 1.4%, while the genotypic TT frequency for MMP2 was 1.8% in the users, with p-value 0.417 and 0.943 respectively. For TIMP2, 78.6% in non-users, 65.5% in users for the GG. For the GC TIMP2, 21.4% versus 32.7% amongst non-users and users, while CC was nil in non-users and just 1 in users, with p-values 0.139 and 1.0. So a lot of complexity at skewing of the data sets in MMP2 and TIMP2 gene polymorphisms amongst tobacco users and non-users in HPV-susceptible cervical cancer risk-mediated cohorts, HPV-mediated cervical cancer susceptible cohorts of Asian Indian women. So we may replicate the findings for multicentric, multi-cohort, pooled population pools of women of varying genetic landscapes, creating a very large population cohort pool and studying MMP2, TIMP2, toll-like receptors 2, 3, 4, and 9, COX-2, which is the cyclooxygenase 2, the autophagy, even apoptosis, along with TGF-beta, may be many other biochemical cell signaling pathways mediated cell signaling components using the single nucleotide polymorphisms, causal genetic association, epidemiology-based studies with statistically powered more than 80% sample size for deriving evidence-based, cost-effective, predictive, and prognostic biomarkers for eventual women's health care and addressing the ever-expanding public health global challenge of gynecologic oncology with emphasis on HPV-mediated cervical cancer and oncofertility. Next slide, please. The study had few limitations as well as loopholes along with study strengths, which included limited sample size of 400 that might have led to a relatively lower statistical power in the subgroups, and the HPV status of the subjects enrolled in the study was unknown, so we did not have access to the tumor tissue samples and were not able to successfully perform a detailed cancer FIGO stage 1 versus stage 2 versus stage 3 versus stage 4 stage-specific molecular and cellular expression study at the mRNA and/or protein level in tissue biopsy samples of cervical cancer due to the constraint in finances, limited financial resources. So we conducted an exploratory public health-oriented HPV-mediated cervical cancer causal genetic association study in a specific population pool of Asian Indian women of North Indian ethnicity. Another study limitation was the lack of availability of established cervical cancer cell lines, for instance, the well-known HeLa cell line that would have further helped in arriving at more definitive conclusions regarding the role of MMP to TIMP2, as well as TLR2, 3, 4, and 9 polymorphisms in cervical cancer in Asian Indian women. Next slide, please. Conclusions. Overall, my innovative study demonstrated lack of association between MMP2 and TIMP2 gene polymorphisms and cervical cancer susceptibility in women of North Indian ethnicity. HPV genotyping suggested that higher the viral load in terms of test cutoff value, higher was susceptibility to develop cervical cancer in the study population of Asian Indian women of North Indian ethnicity with a statistically powered sample size of 400 study subjects, and HPV viral load genotyping may help identifying women at risk of developing cervical cancer